I just want to encourage you again that either if you feel like you'd like to do better in the course or if you'd like to expand on topics that have interested you, to take advantage of the times that the TAs have set aside. They're both very capable people who have a lot to offer, and you can master material better in chunks over time rather than just the week before the test. So do think about setting aside some time to take advantage of that resource. Do people have any questions about Monday's lecture? Okay. Well, then let's return to, oh, by the way, that PowerPoint I was just running is something called Powers of 10, and I will have that on the website if anybody wants to look at it in a little more detail. All right. So are there any other questions about the material we talked about on the membranes up until this point? Okay. Yeah. Sorry. Oh. Yeah, Life of the Cell is on the website. It's on one of the links on the links page. So if you haven't been exploring the links yet, almost all the way to the right-hand side of the toolbar, this thing says links. And in there is where the link to Life of the Cell is. There's both a version with just music, which is beautiful, and there's a version with discussion, so you know what the things are if you don't recognize them offhand. Yes? Do I know anything about the practice? What about the practice exam? When I'll be posting it? I typically post the practice exams about a week before the test itself. So, other questions? Do remember, if you've got a question, usually someone else does too. All right, well, let's go on. We talked a little bit about the role of the glycolipids in creating a coat around cells. That's, I think we need to bring the chattering down a little bit there, guys. Thank you. So far, we've been talking about the membranes as structures without really looking more closely at how membranes can accomplish this function of being selective, about keeping the outside out, the inside in, and the right things moving across. It's really a major role. Some of the genomes that have been examined have as much as 15% of their genes are involved in transport. So this is really a big deal for cells. And broadly speaking, there are two ways in which things get into cells. They're categorized in those broad ways as either passive transport or active transport. In the case of passive transport, something is flowing downhill. That is, there's more of it outside the cell, and it just flows into the cell passively by the law of mass action because there's more on one side than the other. And passive transport can take either of two forms. It can be just plain old diffusion. Good. I'm glad it stopped. There is simple diffusion in which the chemical properties of the compound are such that it can pass through that interior hydrophobic layer at a sufficient rate for the cell to get some of it. So that, for example, steroid hormones just passively diffuse into cells. Oxygen just passively diffuses into cells. There are other things where there's more of it outside the cell than in, but it's not something that's going to very easily pass through that hydrophobic inner component of the lipid bilayer. In that case, we can see something called facilitated diffusion. In the case of facilitated diffusion, there's a transmembrane protein of some kind that allows selectively for certain things to move from the outside to the in, even though they're hydrophilic. So it's still diffusion in the sense that it has to flow down a gradient, but it's been facilitated by the fact that the integral membrane protein permits something hydrophilic to go through a region it shouldn't ordinarily go through. Both these forms of passive transport are in sharp contrast to active transport. And under active transport, you're creating a gradient, or you're reversing a gradient. So in active transport, something that's fairly rare outside the cell is accumulated inside the cell. And since that's not energetically favorable, there has to be an investment of energy into it, usually in the form of ATP. And that's what makes it active transport. So we'll look a little bit at how each of those things happens. Oh, thank you. So we'll take a look at some passive transporters. Porins are a whole family of molecules. They are able to let small molecules go through, and they are somewhat selective, but not terrifically selective. And they give us a sense of the deep relatedness of the tree of life because we find porins in E. coli and other bacteria. We also find them in the outer leaflet of mitochondria and of chloroplasts. And so this is a solution to the problem that was invented a very long time ago. And they have recognizably similar tertiary structures. So this is one shown now in the ribbon diagram. You can see a lot of beta pleated sheets. Each of those ribbons is a beta pleated sheet lined up next to the neighbor. And if you put enough of them together, you get this kind of barrel-like structure, which in the ribbon diagram suggests the presence of an interior pore of some kind. And then when you get a little bit closer to a space-filling diagram, you can see that it's not quite as big a hole as the ribbon diagram suggested, but there is nonetheless a passage for transit through the membrane. And the side chains of the amino acids, residues that are sticking into that area are going to be hydrophilic side chains. The potassium channel is an extremely important membrane transporter. I mentioned briefly before that sodium and potassium are regulated in extremely careful degrees. So there has to be some way for sodium and potassium to move because they're so charged. And if we look at the ribbon diagram for the potassium channel, now we see alpha helices. It's not a beta barrel. It's sort of almost like a funnel of alpha helices that span the membrane. Again, they go from the outer surface of one leaflet to the outer surface of another, with turnarounds. So you can see in the case of this red one, a turnaround that brings it back into the membrane. And if we then look at the channel in the interior, its profile looks something like this. And it's a wonderfully selective channel. A potassium channel will pass 10,000 potassiums compared to the sodiums it will pass. So it's a very selective channel. And this is kind of a surprising thing because potassium is a bigger atom than sodium. So you have a funnel that can let the big thing through and keeps the small thing back. Strange. So how does that happen? Well, it turns out that the potassium channel has, pointing towards its interior, various carbonyl groups that can help strip water off by competing for those hydrogen bonds. And so the potassium can pass through as a naked potassium. But because sodium is smaller, its cloud of water that's in hydrogen bonds with it doesn't get them all stripped off, and now it's kind of stuck and can't get through the hole because it's got this cloud of water molecules that makes it effectively bigger than the naked potassium atom. Very sneaky strategy. Some of the channels in the membrane are gated. That is, they are there for facilitated diffusion. They're just going to let things run down an existing gradient, but they won't do it all the time. Some way or another, they're turned on or turned off. In that case, for instance, you can have a drop in pH. You can have a change in voltage. You can have the presence of specific ions like calcium. One of the niftier things that can signal to these channels is stretch. That is, we now know that there are channels that respond to the fact that the membrane is being stretched. And that's almost certainly mediated partly through the cytoskeleton. So here's another example of how the cytoskeleton and the membrane are in intimate and functional contact. The mechanistic basis for those gated channels takes a variety of forms. Here's one schematic for what's sort of a cap, where in one configuration, that cap is plugging up the channel. in a different configuration, something has caused this to swing away or be relaxed or whatever, and now that central channel is free to pass whatever it is it's supposed to pass. There are also changes in configuration like this, where in one state, the helices of it or whatever subunits, whatever secondary structures are packed in a tight way, and with just a little shift, they rotate enough away from each other to create a channel which now permits the passage of the appropriate molecule. This is an example of how a passive glucose transporter works. In the case of some of these things, they can actually change their shape and in such a way that changes which face is going to be open towards the exterior. So if we start off here with, this is, for the case of this diagram, this is a plasma membrane or cell membrane, and the transporter is open to the outside, to the extracellular space. A glucose molecule can bind to it. Having bound to it, it triggers a shape change. So it's now facing towards the interior. The glucose is free to dissociate into the interior. In the absence of that glucose, it flips back to its previous conformation, and it's ready to take another glucose molecule. If you think about it, this works as a directional transport because there's a gradient. That is, if you had more glucose inside the cell than out, it would end up being a way to get glucose out of the cell. Again, in this case is mentioning that 10% of bacterial genes, depending on the genus, I've seen 15 for some genuses or some species. Another thing about these transporters that accomplish facilitated diffusion is what they carry, how much they carry. Transporters that carry just one molecule, just one way, are called uniports. Port for carrying one thing. So a uniport can just bind one thing. Other transporters, in fact, are specific to two compounds and are always going to carry two compounds in any transport event. But the way they carry them can be quite different. Here's a schematic for a uniport, which just is, one thing going somewhere. In the case of having two ligands, they can either be going in the same direction, which would be symport, like sympathy, feeling the same thing with someone, or they can be going in opposite directions, in which case you have an antiport. And both of these strategies are used. So that was all passive transport. All those channels and stuff were examples of facilitated diffusion. What about active transport? Very expensive part of the life of the cell, that at least some things have to be maintained against the gradient in order to be a cell. So that energy is invested that way. We're going to see how maintaining a gradient of one thing can allow you to create a gradient of something else. And the first example we're going to look at for that is one of the more important active transporters, which is the sodium-potassium ATPase. It got this name because it was a membrane protein that would consume ATP in the presence of sodium and potassium. So it was identified as a component of membranes before it was appreciated that it was actually a transporter. And its role is to make sure that sodium and potassium are on the right sides of the membrane. And the right sides of the membrane, again, are potassium to the inside of the cell, sodium to the outside of the cell. That's the ongoing state that most cells want to maintain. It's not an it's not isoelectric. It's an exchanger that will exchange three sodiums for two potassiums. So in addition to creating a gradient of sodium and a gradient of potassium, it will also be creating an electrical gradient as well, because the electrical charge is not conserved during this. So for every two potassiums it takes into the cell, it's kicking out three sodiums and losing one net charge. And each cycle of it costs just one ATP. So for one ATP, you can do quite a lot of work. We're going to see that its mechanism is kind of nifty because it uses a phosphoaspartate, kind of a sly trick. So let's start at the beginning. here we have the sodium-potassium ATPase at the it's a cycle, so we could pick it anywhere, but let's just start here. Open towards the cytosol, towards the cytoplasm, and three sodiums combined in that interior space. When ATP binds, that will trigger a change in conformation that transfers a phosphate from that ATP onto an aspartate of the ATPase, the sodium-potassium ATPase. And with that, there is a change in conformation. The sort of, you can visualize it in your minds if you like as kind of a flip. And now the sodium ions are free to enter the extracellular space. That was this picture here. Once those sodiums have left that part of the transporter and are exposing that face, naked to the extracellular space, potassiums are free to bind. And if potassiums bind instead of some sodiums rebinding, because if you have to think logically, it could be that sodiums would rebind. That won't accomplish anything. But if potassiums bind at this point, it will trigger a release of the phosphate group from the aspartate. And with the loss of that aspartate, there'll be a change in conformation, so it now faces back into the interior again. And the potassiums are free to dissociate back in the cytoplasm. And that puts us back where we were at the beginning of the cycle. So it's a way to use the energy of an ATP, but not as a part of the direct reaction. It's as a way of favoring or not favoring particular conformations of the protein. Now, cells can be very sneaky and use this gradient of sodium and potassium to move other things. And this is called indirect active transport, because it's something that will only keep on happening as long as there's some ATP available to maintain that gradient, but the transport itself doesn't use up the ATP. And I'm just going to explore an example of that here with you. So in this diagram, what we have here is a molecule of the sodium-potassium ATPase. It's consuming ATP, and in the process, potassium is being pumped into the cell, sodium is being pumped out of the cell. So at a steady state, as ATP is being steadily consumed, there'll be a higher concentration of potassium inside and sodium outside. But in that same cell membrane is a molecule, an integral membrane protein that's adapted for facilitated diffusion. And this one happens to be a symport that can take sodium and glucose at the same time. And so the sodium will bind because there's more sodium outside than in. Glucose binds, and the two go together into the interior. And then we can keep that cycle going because the sodium that came in will be pumped back out at the cost of ATP. This part is facilitated diffusion, but it's called indirect active transport because it would only keep going as long as there is an ion gradient across the cell. Any questions about that before we go on? Now, this particular cell happens to be an intestinal cell. It's part of the specialization of the gut that's involved with absorption. And the long-term goal is to get that glucose out to the body. And so the rest of this transport mechanism seen from a high level is a second glucose transporter. And this is one that is just a plain old facilitated diffusion type transporter. It's a uniport that just carries glucose from one side to the other, and it just does it from whichever way the gradient is. And since using the ATPase here, you're going to be able to actually pump glucose into the cell relative to the environment, now you have a gradient where the glucose is higher here than there, and that transporter permits that glucose now to simply diffuse out into the circulation where it can be carried to the liver and the rest of the body. So this is really a three-component system that requires ATP to get glucose from the gut into the bloodstream. Any other questions about that? Okay. Another place where ion transport is extremely important is in the whole domain that's called membrane potentials. Now, membrane potentials were first really understood in the context of nerves. But you have to keep in mind that nerves are not the only kind of cell that can generate membrane potentials. They're just the most dramatic ones and the ones where it's easiest to see the spatial consequences of it. But you remember I pointed out that the sodium-potassium ATPase generates an electrical gradient as well as an ion gradient. And so the way a nerve impulse traveling works is something like this. Let's say we have some kind of a stimulus on the cell. And that stimulus, whatever it is, it might be a neurotransmitter, it might be touch, it could be any of a number of things, stimulates a sodium channel to open, and that sodium flows into the cell passively because there's a gradient. And as that sodium flows into the cell, it's going to neutralize the electrical difference that was in the cell to begin with. So in this initial state, the cell had a minus 70 millivolt potential difference, and the stimulus is going to trigger sodium influx. As that sodium comes in, here you can see the local voltage has risen to plus 50 millivolts. It's actually reversed the cell membrane potential. Well, with this change in the voltage, that can then trigger voltage-sensitive potassium channels. Remember, there's more potassium inside the cell than outside, and in this case, there are gated membrane carriers that are sensitive to voltage, and they now open and let the potassium out. And the potassium will continue to flow until it restores the voltage difference, because that's the key or the signal to the potassium channel. So what we've seen in this part of the diagram so far is the stimulus here creates a fluctuation of sodium and a fluctuation in potassium that then restores that potential difference. Now, what's kind of cute about that is that the change in the polarization of the cell will also trigger other sodium channels and potassium channels down through the rest of that axon. So this shift in cell voltage will pass as a wave down the axon. And the reason it passes directionally in one direction and not in both directions is because of the tendency of the channels to be refractory for a while. They won't respond for a little bit. They have to kind of rest and come back into be responsive. And so the net effect of this is for an electrical impulse to pass from one end of the nerve down to the other. So, questions about that? Okay. We see this especially dramatically in a class of nerves called the myelinated nerves. In this picture, we have a transmission electron micrograph where we've cut a thin section perpendicular to the long axis of the neuron. And you can see here, this is the cytoplasmic part of the axon, which is pretty small and doesn't have a lot of dramatic stuff in it. And here we see layers and layers of myelin around it that were deposited around that by another cell. It acts as insulation, and so instead of having to travel completely down every part of the neuron, the electrical impulse jumps from node to node. And the myelination allows both for faster conductivity, so like 20 times faster, and also, to some extent, specificity. It's kind of interesting that this myelination process is a very slow one, and it is still continuing up to 20 to 30 years of age. My husband calls the little stage around this tall when you're kind of figuring out how to walk and talk, the software development stage, and there's something to that. But the fact that we continue to myelinate our nerves means there's even a hardware component to our brains for quite a while. Now, I mentioned briefly that one of the things that might have triggered that impulse on the nerve was a neurotransmitter. Maybe there was some kind of a molecule that triggered that sodium channel. And in fact, one of the common neurotransmitters that will trigger the sodium is acetylcholine. And the ability of nerves to talk to each other at the synapse has been taken as a model for membrane fusion because so much of it happens in such a local area with neurons. And what we've learned from nerves has, in fact, proven useful in other contexts, as in so many things in biochemistry. Part of what, sort of some of the stages that go on during synaptic transmission, the passage of a signal at a synapse between one nerve and another is the release of some kind of a neurotransmitter, which will involve membrane fusion, reuptake into the cell, and often cases of then degradation either outside the cell or inside the cell. Here we have one of these neurotransmitters. This is acetylcholine. There's the choline ion there. It's been acetylated. You may recognize acetylcholine because it is the neurotransmitter that may play a role in Alzheimer's, and there are anti-Alzheimer's drugs that prolong the life of acetylcholine. And what keeps the acetylcholine from doing things forever and forever is that in that gap in the neural synapse, there are enzymes, acetylcholinesterase, that can break down the acetylcholine into an acetate group and a choline molecule. This is a particularly interesting enzyme because some of the neurotoxins out there, which are, for instance, insecticides, are inhibitors of this specific enzyme, acetylcholinesterase, and also some poison gases. I believe the fugu fish, do you remember? Does the fugu fish inhibit acetylcholinesterase? Or am I thinking of something else? Okay. So let's take a little look at that in some detail. What does that look like when it's actually doing itself? And this is the structure we're going to be working from. So we've had an action potential. This action potential has been going right on down the axon, but nerves don't stretch clear through your body. They have to, connect to something else to pass the message on. So the action potential continues down in the way we described from the sodium channels and potassium channels until it gets here to a terminus. This is the end of the axon or axon terminus. And that change in the voltage is going to trigger calcium channels now. Another aspect of the ionic life of a eukaryotic cell is that calcium is more abundant outside the cell than in the cytoplasm. The cytoplasm itself tends to be submicromolar amounts of calcium and substantially more than that outside the cell. So that means that calcium can be used as a signal to do something or to stop doing something. I believe, and I'm going to be corrected on this, that there are no prokaryotic things that use calcium as a signal. Sorry. Yeah. So this is an invention of the eukaryotes, and it gets used in a very sneaky ways. So we've had this action potential come down. It's triggered this calcium channel, so calcium is now going to flow from outside the cell to inside the nerve terminus. And this space here between the nerve terminus or the axon terminus and the muscle cell that's going to be addressed or spoken to is called a synaptic cleft. The synapse means joining together, a place of contact, and they didn't know when they named it that there's actually a little space. So this is the synaptic cleft. There are membrane-bound vesicles, which get called synaptic vesicles. And what happens when the calcium flows into that axon terminus is that it causes some of these synaptic vesicles to fuse with the membrane here, and you can sort of see that diagrammatically. And as they fuse with the membrane, they're dumping acetylcholine into that synaptic cleft, a very small space. There are acetylcholine receptors on the nerve cell, and these, again, can be thought of as sodium channels. And so for a brief period of time, the acetylcholine is there and can trigger a signal, but the release of acetylcholine is just for a short period, and whatever is left in that gap is going to be cleared out by acetylcholinesterase. And so that then provokes the signal for muscle We're starting to understand some of what goes into membrane fusion at the level of molecules. Some of the things that were maybe first observed at the synapse, in fact, have been found to be more common than that. And some of the proteins that are involved with this have been identified. One group of proteins is called snares. If you ever become a protein chemist, part of your privilege is you get to name things with cute names. So it's always important to find a cute, memorable name for things. Snares stands for soluble, NEM-sensitive attachment protein receptors. So they were soluble once they were released from the membranes. They're NEM-sensitive, which means that there's a small side chain modifying compound that was used to probe their behavior, attachment protein receptors. So these are the things that are going to be involved with the contact between the two. So here you have a vesicle with its set of snares. You have a plasma membrane with a different set of snares. And when they become close enough, they can then form coiled coils. In this case, there's 4 alpha helices that are bundled together. This is a 4-helix coil. And then that provides a sort of a clamp, if you like, that holds those 2 membranes close together. In the absence of that 4-helix bundle, these things are they're kind of close, but they could come and go if you like. Once they come close enough for that bundle to form, that then stabilizes the vesicle close to the membrane. When we look at membrane fusion in a little more detail, if we go in and hone in on it down to the level of leaflets, not just treating the membranes as a single line, but appreciating the fact that all these membranes have 2 leaflets, it starts to give you an appreciation for the fact that there's some fairly complex stuff going on at the level of molecules. So here we have a vesicle. We're not showing the snares in this case. And with 2 leaflets, with your charged heads on the outside, your hydrophobic stuff to the interior, and a relatively flat plasma membrane. It's flat relative to the vesicle, at least. The snares can allow them to come as close as this. And if you think about it, this area where the 2 have just sort of touched the very point of contact, that's the polar stuff. Those are the polar head groups interacting, which might be the same charge or they might be of different charges. It depends on what the cell has made those membranes out of. But they are both hydrophilic. We can then actually see these following stages of rearrangement of the leaflets where having made this sort of kissing contact with the polar parts, small amounts of rearrangement can generate this sort of crossover configuration where now it's the hydrophobic parts that are in contact with each other. And with continued rearrangement of those and with these 2 pinching off, you now end up with a continuous space to the outside of the cell. So you've turned something that was topologically inside the cell here into something that's topologically in contact with the outside environment there. But one of the things that should be clear looking at this is that in order to accomplish all of this, there has to be some way for the cell to control membrane curvature, how tightly curved they are, what would be the radius of a circle that would overlap with that. Because here you have a very low curvature thing, and here you have very high curvature things. Somehow or other, you've got to adjust that. And certainly one of the ways of doing that is by actually changing the membrane lipids. You remember we talked about how the number of tails that something had affected what you could make out of it. When you have a phospholipid with 2 acyl tails, it's going to make the flat kind of thing. And if you have an admixture of things that have lost one of those tails, it's going to tend to make things more curvy. Head groups are another thing that can change the curvature of the membrane. This is another transmembrane complex that's pretty nifty in that it's what's responsible for the fact that water can get into cells. We take water for granted, but it, in fact, can't freely diffuse through a membrane bilayer because that interior is hydrophobic. Here's a ribbon diagram for a single subunit of aquaporin. You can see it's got multiple alpha helices, and 4 of those together make kind of a hard to appreciate side view. If we do a space-filling transformation on that diagram and then look down from on top, we can see the tetramer looks something like this. The water channels are in each subunit, like that. The little space to the center is not the place of transmission. So what would that look like in a little more detail? Here we're now looking at a single subunit. You can't really appreciate the alpha helices very easily here because of space filling. But what the space filling diagram can do is highlight for you the structure of that channel. And if you look at the channel here, it's kind of funny. You have this side of it is hydrophobic. And there's just this sort of string of asparagine residues that are lined up down that pore. This is a very nifty pore. It won't pass urea, which here's the structure of urea, a polar compound, you know, not that much larger than water. It won't pass protons, which are actually smaller than water. It passes water. That sounded bad. It permits water to pass through. And so the way it accomplishes that is that the asparagine can interrupt the chain of hydrogen bonds. You remember back to the structure of water, of each one having 3 to 4 hydrogen bonds. What happens here is that the asparagines pluck off some of those hydrogen bonds so that now this water molecule is no longer a part of that whole network. It's still hydrogen bonded, but it's hydrogen bonded to the asparagines and can kind of swivel its way down through the aquaporin and get down to the cytoplasm. This is a little taste for you of what's possible now through the computer calculations. Here they've attempted to calculate in more detail the positions of the various things. And in particular, take a look. Let me see what your black and white versions look like. In particular, it's not really clear in the gray versions. Sorry about that. So watch with me on the screen then. Here you have the little string of blue water molecules. And you can see how it's a single file of water molecules threading their way through the center of the aquaporin. I'd like to close off then with a few sort of working examples of membranes and work through them with you. Take a look at this diagram. And in this diagram, there are these 6 things. And think through first for each of them what you think, which letter would be appropriate. And you can chat with each other while you do that if you like. Mm-hmm. Let's see if we've gotten some consensus on a few of them. Do anybody want to take a stab at which of these, if any of them, might be a glycoprotein? A? Would anybody want to explain why? Why would A be a glycoprotein? Sorry? I'm sorry, what? Okay, so in other words, it's extracellular, and that's probably the more important dimension than being peripheral. And the sugar parts are always are pretty much exclusively exposed to the outside. It's the good bet. It's the way to bet on the horse race. Which of these proteins might be removed with salt? Either chip in or hand up or either one. Which ones might be removable with salt? I know some of you know. Sorry. Someone was suggesting A. Okay. Is there anything else that might be removed with salt? A and C. So both A and C, peripheral, could be removed with salt, very likely. But then which one would need strong detergents? D. Okay. And D would need that because... Why would D need strong detergents? Anyone want to offer an explanation? I'm sorry, a little louder? Right. D is an integral membrane protein. It's got a lot of hydrophobic contacts into that lipid bilayer hydrophobic region, and only a detergent is really going to disrupt that. The salt could disrupt the ionic interactions for the peripheral ones, but it needs detergent for the hydrophobic contacts. Which of them might be a sodium transporter? Okay. D is the only integral membrane protein there, so it makes a good transporter. Which of these might be ceramides or gangliosides? Anyone want to venture? The ceramides and gangliosides are among the membrane lipids that have sugar components to them. And so that would be an extracellular face where you might expect to see them. Which of these might flip-flop with a translocase? Did I hear E? Anybody want to explain your reasoning? Okay, so the thinking here is that membrane lipids in general would need a translocase in order to flip at all. And both E and B are membrane lipids, but D, with the glyco residues on it, is probably not going to flip back inside. So the candidate here would be E. Okay, and everybody comfy with that? I'm sorry. Let me back up here. A 5. A ceramide or ganglioside would have been B. Those are membrane lipids that have carbohydrate components to them. So what kind of anchors do these membranes, these proteins have? Anyone want to give a venture on this one that looks kind of simple by comparison to the others? So that's going to be something like myristate or palmitate. It's a simple fatty acyl tail. What about this one? This is one of the prenylated membrane lipids. The first one is a simple fatty acyl tail. It's going to be either probably myristil or palmitoyl, one of those. You'd have to count the carbons to get it right, and I don't think those are countable carbons. Those look like hypothetical carbons. So this is a prenylated tail here with the branched hydrocarbons, an aromatic, not aromatic, the branched unsaturated acyl tail. And here's one of the ones that's both got a sugar and the phosphate. This is a phosphonothietidal glycolipid. So it has the 3-carbon backbone here with its own fatty acyl tails, and then the protein is actually linked through this large exterior conglomeration that includes a lot of carbohydrate. This kind of a connection between a membrane, so this is the protein over there, and this is all this stuff. One advantage of this kind of ornate carbohydrate as an anchor is that it allows for this protein to be detached from the membrane fairly simply, that there are examples where you have a protein that is sometimes anchored to the membrane and sometimes is attached through one of these things, and you can shed it very quickly depending on the circumstances. Okay. I think I'm going to skip this one for now. There's more details in the book if you want to read through it. Let's take a look at panel 44. Take a look at those structures and chat with each other if you want to think of what would be their relative passive diffusion rate through a membrane. In the absence of a channel, in the absence of active transport, what would be their relative diffusion rate as simple passive diffusion? Anybody want to venture which one would probably go through the most quickly? Did I hear anything for our candidate for Speedy Gonzalez on this? Yes? Okay, so in other words, you're proposing that ethylene glycol would be the one that would pass through the membrane the most quickly, and your reasoning for that? Right. So in other words, it does have alcohol groups, but those alcohol groups are not going to be as polar as the carbonyl oxygens. So this has sort of a little bit of, carbon, hydrogen stuff in the middle and just the alcohol groups. Do you want to take a stab at some of the reasoning for the other compounds? So you were saying that this would be the slowest of them then? I suspect not. If you look, let's look at a little bit of structural analogy here. So this has the group, and this has this group, and this has this group. So these three things are going to be similar except for the effect of the other parts of the molecule, right? So we can cancel those out of the conversation, so to speak. The butyramide has a 3-hydrocarbon chain. The acetamide has a methyl group, and the urea has an amide group. So this would probably be more diffusible. Remember that the interior of that membrane is hydrophobic, and so that tail makes it more hydrophobic. Ionic things almost never freely pass through a membrane. Polar ones will go through less well depending on how polar they are or what their balance of polar and nonpolar is. So this has a longer nonpolar tail than this one, and this is more nonpolar than that. So that would probably be the quickest, next, and then last. And I think it's time to say goodbye for now, and I'll see you all Friday. Hi, what's up?
This would actually be